Snakebite remains a serious yet neglected public health issue in India. With Snakebite remains a serious yet neglected public health issue in India, with thousands of lives lost each year. But now, landmark research is changing how we understand and treat venomous bites. Scientists have for the first time decoded how the saw-scaled viper produces its venom at the genetic level. In a parallel breakthrough, researchers have also found that oral pills, rather than traditional antivenoms, could be a game changing treatment for bites from the deadly Russell's viper, responsible for over half of India's snakebite cases. On this note, I, Dr. Anubha Jain, senior journalist from ETV Bharat, Bengaluru, will discuss these interesting research studies with the Associate Professor Kartik Sunagar from the Indian Institute of Science, Bengaluru, the lead author behind these groundbreaking studies. Welcome to our channel, Professor Sunagar.
Thank you.
So first, we would like to know about your research work in Equus carinatus or saw-scaled viper, and the second one, Russell's viper study. Over to you, sir.
Sure. So basically, in the saw-scale viper study, we looked at how the snake produces its venom and how it regulates it by sequencing its transcriptome. Basically, the genes, basically how genes are translated into toxins. That's what we decoded here. And as you mentioned, in another study in parallel, we actually looked at Russell's viper, which is the clinically most important snake in the world, in the sense that it kills and it causes amputations of a lot of bite victims, more than any other snake species anywhere in the world. So we actually screened a couple of drugs. These are small molecule inhibitors, which pose several advantages over conventional antivenoms. And we showed how this drug can actually single-handedly neutralize venoms of rhesus vipers from various corners of India.
Okay, right. So you discovered a novel protease inhibitor in the saw-scaled viper venom. Could you explain its significance and how it might contribute to the venom's potency or medical complications?
So soft-scale viper venoms are, rich in proteases. So these are enzymes that degrade other proteins, right? So that's why when soft-scale viper bites somebody, you see necrosis and other symptoms, affecting the blood and so on. All of that happens because of these proteases. So you can think of it, right? When the snake produces these proteases in its own venom gland, how does it protect itself? Right? So that's why it produces also this inhibitor, which binds to these proteases properly and then ensures that the venom gland itself is not auto-digested, and it only gets activated once the venom is injected into the prey or accidental human victims.
Mhm. Right. So, given the observed effectiveness of commercial Indian antivenoms in your study, what do you see as their, current limitations, and how critical is the need for region-specific or population-tailored antivenoms in India, considering the genetic diversity in venom profiles?
Okay. So answering the first part of your question, conventional antivenoms are produced by immunizing horses, right? So you basically collect snake venoms and then you inject them into horses. Horse body will now produce antibodies against this venom. So you purify those antibodies and then administer it into snakebite victims. The first problem itself is that you are harming animals by immunizing them with venoms, right? So there's a lot of toxicity that you can see in those horses. So that's first. The problem to the human is that these are horse antibodies, right? So you're injecting horse antibodies into humans. So the human immune system recognizes these as foreign antibodies. So it can mount a response against these antibodies themselves. So the antivenom itself can be dangerous. The third is that we are using a technology that is over 100 years old, right? So the amount of antibodies against snake venoms in the horse sera is very, very low. So you require many vials for treatment. That increases the cost as well as the risk to these adverse reactions that I just mentioned, right? Anaphylaxis and other things. The antibody itself can, harm the bite victim. So these are some of the disadvantages. And coming back to your question about regional antivenoms, so another disadvantage of conventional antivenom is that because it's a 100 years old technology, it only produces antibodies against, the snake population that you have collected for antivenom production. For example, for treating snake bites in India, all the venoms are collected from just two districts in Tamil Nadu.
Mhm.
So the venoms are collected from the four species of snakes there, and that is sold to the manufacturers. Manufacturers use it to produce antivenom, and this antivenom is used for treating snake bites all across India. The problem is we have extensively shown that different populations of the same species of snake can produce very different venoms.
Mhm.
So when this happens, this antibody or antivenom will not be able to neutralize those newer venoms, right? So for example, let's say we have now, we have shown this all across India, but just to give you an example,
Mhm.
although Karnataka is a neighboring state of Tamil Nadu, we have shown that the antivenom doesn't neutralize the venom very well here itself. So that is because of the extent of venom variation in snakes, right?
Mhm.
So that's why you require regional antivenoms, which is another study that is happening in the lab. So we have produced the first regional antivenoms for
Mhm.
Western India, and we have seen how. So basically, what you do is you collect venoms of the local populations of snakes here,
Mhm.
and then produce a specific cocktail against those populations. So that's what is a regional antivenom. And there also we have made some progress.
Okay. So the inhibitors like
Yeah,
Veresplidin and merimastine, so both
yeah,
repurposed drugs, so have
correct.
shown promising results in neutralizing Russell venom. And how do they work
Yeah,
differently from traditional antivenoms? Can you please elaborate?
yeah. So the so I'll give you advantages of small molecule inhibitors. I think that's very important to understand. So the first one is, as you said, these are repurposed drugs, right? So we are repurposing them for
Mhm.
snakebite, meaning that they were originally made for treating cancer and heart diseases. But now
Okay.
we are trying to see whether they can also be useful for snakebite because they target very similar enzymes
Mhm.
in snake venoms, right? So the advantage is here is that one of the major advantages is that you can take them as pills, right? So you don't need to go to a hospital.
Mhm.
As soon as you're bitten by a snake in the wild, you could just take these pills like crocin tablets. You
Mhm.
don't need a doctor to help you to take those tablets, right? And yeah, so that's the first major advantage. The second advantage is that unlike antivenoms, this can work broadly, meaning that as I told you earlier, when antivenoms produced in Tamil Nadu may not be very effective in Karnataka, right? But we have shown that these antivenoms actually work all across India. Sorry,
Okay.
these small molecule inhibitors, they work all across India. We tested them against Russell Swiper populations from almost every corner of India, and we show that various combinations, either one of these drugs or both of them might be required to treat snake bites in many of these places, right?
Mhm.
So that's the second advantage. The third advantage is you're not immunizing animals anymore, right? Because these are chemicals, these are synthetic chemicals. So you are reducing that harm to animals also.
Mhm.
And finally, of course, because these are chemically made, they will be a lot more cheaper than conventional antibiotics. And they don't really mount an antibody response. So you don't even have those problems like anaphylaxis that you see with current antivenoms. So in all the problems that I told you about conventional antivenoms, these drugs can address each one of those problems.
Okay. Wow, wonderful. So, do you envision a future where snake bite treatment begins in the field with pills and is followed by antivenom if needed, a hybrid model?
Yeah, I think you hit the nail on the head. That's exactly what we are trying to address now. So we are going to work with ICMR
Okay.
to basically clinically test these drugs in humans
Mhm.
and see how effective they are. And if they clear clinical trials and if we see that effectiveness, then yeah, as you absolutely correctly pointed out, either they will be used as adjunct therapies, meaning that you start with small molecule inhibitors and reduce the dose of antivenoms needed. Or if you see complete protection in humans as well, then you can completely replace conventional antivenoms and just administer these small molecules.
Okay. So, since these are orally available drugs, what advantages will they offer, especially in India's rural or under-reserved regions compared to injectable antivenoms?
Yeah, so as I said earlier, because these are the conventional antivenoms also don't require refrigeration. That's a very common misunderstanding
Okay.
because many of the conventional antivenoms are made in a lyophilized form, meaning that they are freeze dried, so you can
Mhm.
store them at room temperature also.
Okay.
But some clinicians also rely on the liquid formulation of the antivenom. So if it is made in the liquid form, then you need refrigeration, right? So in terms of that, this is very easy because this doesn't require refrigeration. They are very stable at room temperatures,
Mhm.
right? So that's the first advantage. The second advantage, again, as I said, is the most bite victims of snakes are farmers and laborers, right? So it's a poor man's problem. That's why it's one of the most neglected of the neglected tropical diseases. Nobody worries about them because these are poor people who get affected by it.
Yeah.
So the advantage of small molecule inhibitors is that these are going to be very inexpensive for them, right? Um, Antivenoms are free, but only if you go to government hospitals.
Mhm.
And a lot of bite victims can't go to government hospitals or are probably sent to private hospitals too. So there you have to pay for antivenoms, and then you also have to pay a lot of money for antivenom for snakebite treatment, right? But with small molecule inhibitors, all of those associated costs will go down if they are very effective in neutralizing, the major toxic symptoms of bite.
Mhm. Right. So, what are the next steps? If I want to ask, what timeline do you envision for bringing these drugs into mainstream use?
Okay, can't really talk about timelines because these are all, there are lots of regulatory approvals that are required, and a lot of testing still is there. But what I what I would like to point out is that the future for snakebite treatment in India is very bright because
Mhm.
we have multiple solutions. See, one of the things that we didn't talk about are monoclonal antibodies. So instead of making antivenoms in horses, you
Mhm.
can also make these antibodies in the lab. So we have made such antibodies in the lab which are very effective. Again, address all the problems that I mentioned of conventional antivenoms. So there
Okay.
is a lot of testing that is needed for both monoclonal antibodies, for small molecule inhibitors. And yeah, once they clear those trials, then I think it'll be easier to introduce them to market. Small molecule inhibitors can actually go to the market a lot more faster
Mhm.
because they are already cleared, for other diseases, right? For treating other diseases. So we know that they are safe to administer in a short duration. So we already have that safety data. So as long as we see protection against snakebite effects, these can be immediately made available to the market with, as little time as possible.
Okay. One important thing I like to ask, what has been the biggest challenge in driving such a paradigm shift, like from Venom profiling to pill-based therapy? Your research represent a major shift. So, what
Yeah.
has been the biggest challenge?
Yeah, I mean, there are multiple challenges. I talked about all of this in last 10 minutes, but this has probably taken, almost a decade for us to do, right? And that's because, so one of the major problems is that this is very multidisciplinary. You need to do a lot of different types of research. So, and
Mhm.
usually researchers focus on one aspect of these, right? So by establishing all the technologies in my lab here, we were able to, implement all of these different strategies parallelly. So that's why we understand so much about the biology of snake venoms. And using that understanding, we are now making better therapies. The
Mhm.
second problem are obviously regulatory issues. It takes a lot of time to get approvals to collect snakes and their venoms. So sometimes it takes several years. For some regions, we still haven't heard back from the forest departments. So we don't know the effectiveness of the small molecule inhibitors or antibodies in those regions, unfortunately. So if the government pushes for it and helps us to get these clearances, we'll be able to test these newer products against snakes there, and we'll be able to tell whether these solutions will also be useful in those regions. And all of this is absolutely very important, but unfortunately, is very challenging. Yeah. So those are the two major issues I would.
Right. So, in the end, if you have to summarize the potential of your research in one sentence to the Indian public or policymaker, what would you say?
It's very difficult to summarize our research in one sentence, but yeah, I think all these advanced solutions for snake bites can save the lives and limbs of India's 100,000 snake bite victims, because India suffers the brunt of snake bites in the world, right? I mean, over 50% of all the deaths that happen due to snake bites around the world happen in India,
True.
and so I think our research is very important in that translational space.
All right, all right. And thank you so much, Professor Sunagar, and thank you for your time and for your insightful information you have shared with us. Thanks a lot.